Hello, welcome to my coronavirus classroom. I'm Janessa Jacobs, and these are the blood vessels. So when we talked about the heart, we talked about the definitions, our very basic definitions of an artery and a vein. So we're going to elaborate on those definitions today, and we're going to talk about all of our blood vessels in great detail. So as you recall, when we discussed the heart, we said arteries are the vessels that carry blood away from the heart. In the systemic circuit, this blood is oxygenated. In the pulmonary circuit, this blood is deoxygenated. These vessels have a high resistance to blood pressure. They are the ones that have to resist the pressure that's coming from ventricular systole. So arteries are sometimes called our resistance vessels. Our next type of blood vessel are the exchange vessels, the capillaries. These are our smallest vessels, and this is going to be the site of nutrient and gas exchange for the tissues. So this is the site of nutrient and gas exchange, and thus we call them exchange vessels. The last type of blood vessel we have, as we discussed with the heart, are veins, and veins return blood to the heart. So these return blood to the heart. So in the systemic circuit, they're returning deoxygenated blood to the heart to go out to the pulmonary circuit. And then in the pulmonary circuit, oxygenated blood is returning to the heart through our pulmonary veins. And veins have a high capacity to hold blood, so we call them our capacitance vessels. And we'll talk more about why in a little bit. Okay, so our arteries are going to carry blood away from the heart out to capillaries, where we're going to do nutrient and gas exchange. And when we get to those capillaries, on the arterial side, we're going to push out 100% of the material that we're going to push out. And on the venous side, we're only going to pick back up 85%. Which means the remaining 15% of fluid is stuck there in the interstitial space. It gets picked up in these lymphatic vessels. So if we think about the relationship of all of these things to each other, we have the heart, and blood is going to leave the heart in arteries. First, elastic arteries. And then these are going to get smaller and become muscular arteries that are going to lead into these little arteries called arterioles. Our little arterioles are going to lead into this thing called a meta-arteriole, and that is going to drain into our capillary bed. So this is the systemic circuit I'm kind of sketching right now. Our meta-arteriole could bypass the capillary bed and drain right into this thing called a thoroughfare channel. Or we can have capillary, our nutrient and gas exchange, and then drain into the post-capillary venule. So this would be a little post-capillary venule. All our little venules are going to converge to become small veins that are going to converge to become medium and large veins. And then those are going to return our blood to the right atrium. So what are the largest veins? It would be our superior and inferior vena cava. And then our coronary sinus would also be draining deoxygenated blood from the systemic circuit. And then our largest artery then would be the aorta, that's leading away. So this would be opposite in the pulmonary circuit. Then our pulmonary trunk would be the artery that is going to take blood away to our little arterioles in the lungs. And now in the lungs then, at the capillary beds there in the lungs, we would have nutrient and gas exchange and pick that oxygenated blood back up in our pulmonary veins that drop into the left atrium. Okay, now that's the flow of blood and the relationship to the heart. What about this extra 15% of fluid I mentioned? So if we look at the arterial side of a capillary bed, we push out 100% of what we're going to push out. But on the venous side, we only pick back up 85%. So 15% of what got pushed out here would just be stuck out here. You would have swelling on all of your tissues if we didn't have some way to return it. So we do have some way to return it. We have these little lymphatic capillaries that are super cool that you find associated with normal capillary beds, and they're going to become these vessels, lymphatic vessels that run past lymph nodes, and then are going to become these other lymphatic vessels, ultimately these lymphatic ducts that drain into your subclavian veins to return that blood, or to return that interstitial fluid to the blood. So I draw this here because we're going to talk about this for your next exam. So just be aware of that. Now, that's kind of the relationship of all of those structures. Okay. Now we need to look at the generalized structure of our blood vessel walls, and then we'll look at each specific type of blood vessel and see how the differences in the layers of the wall are going to give different properties to different blood vessel types. So the innermost layer of the blood vessel wall that is in contact with the blood is called the tunica intima, or the tunica interna. And this is in contact with blood. And so under normal conditions, this should actually repel blood cells and platelets so that we can have blood flow. So normally repels our blood cells and platelets. Why do I tell you that? Well, because if you have damage, then it can do something else. It can express these things called cell adhesion molecules. I'm just going to write CAMs right now, and we will talk about what they are when we get to the inflammatory response. So in a damaged vessel, we could say our damaged cells, or in damaged tissues, we can express CAMs. And we'll talk about the inflammatory response and how that happens for your next exam, or when we get to the lymphatic system. The immune system, actually. All right. What else? So next, our next layer, so this is our innermost layer here if we're looking at a cross section. Our next layer is called the tunica media, and this is smooth muscle. It's the thickest layer in our arteries. So tunica media, this is smooth muscle. This layer is the layer that responds to the vasomotor center. Where does the vasomotor center originate? The medulla oblongata. And so impulses that control vasomotion, or the movement of our blood vessels, come from the vasomotor center and are going to have an impact on the tunica media of our blood vessels, which is smooth muscle. So we could say this is responsible for vasomotion. And we have two types of vasomotion. We can get wider or we can get narrower. We can vasodilate or we can vasoconstrict. So vasomotion is our movement of our blood vessels. So we could decrease our diameter, and that would be vasoconstriction. Or we could increase our diameter, and that would be vasodilation. Okay, the outermost layer is dense irregular connective tissue. that anchors our blood vessels to each other, our companion vessels, so arteries and veins run together, and it anchors them to each other, and it anchors them to the surrounding structures. It also contains the blood vessels that serve the blood vessels. What? Blood vessels are full of living cells, so they have to have blood supply. It's thought that the innermost layers get everything that they need from the blood flowing through them. But the outer layers, especially in our great vessels near the heart, are really far away from that. So they have to have their own blood supply, and we find that as what's called vasa vasorum in the tunica externa. So the tunica externa is dense, irregular connective tissue. It anchors companion vessels to each other and to surrounding structures, and it contains the vasa vasorum. I'm actually going to write that over here. Contains the vasa vasorum, and these are the blood vessels serving the blood vessels. Okay, so I have a question for you. Which layer of the vessel wall responds to the vasomotor center? A, tunica intima, B, tunica media, C, tunica externa, D, all of the above. You're right, it's the tunica media because it's got the smooth muscle that can respond to nervous stimulation and contract or relax. All right, so something that I need to mention is that we have these two elastic membranes in our larger vessels that we find between the tunica intima and the tunica media. And so this is the internal elastic lamina that we find between the tunica intima and the tunica media, and then the external elastic lamina, which is between our tunica media and our tunica externa. And as we go from our arteries to capillaries, we get smaller and smaller. At first, we're going to go from this high proportion of elastic connective tissue to these smaller arteries that are mostly muscle, to our little arterioles that have some smooth muscle around them. But then we're going to get to our capillary beds and only have our simple squamous epithelium and a small basement membrane. Then in our little venules, we'll start seeing more connective tissue. And then in our small veins, we'll start seeing more of even more connective tissue, and we'll start seeing some smooth muscle cells, and then some layers of smooth muscle, and then as we get into our veins that are 2 millimeters and larger in diameter, we'll start seeing the appearance of valves in the tunica intima. And those valves are going to be really important because they're going to prevent the backflow of blood from one valved segment of the vein to the next, because all of our pressure due to ventricular systole gets dispersed in the capillary bed, so there's different pressures moving blood through veins. And we'll talk about those in a little bit, but I needed to get that kind of addressed before we move away from the structure of our blood vessel wall. So in the tunica intima, we could say in our veins, it contains valves to prevent backflow of blood. And so like when we get down to our little tiniest blood vessels, no, we're not going to have any more internal elastic lamina and external elastic lamina. We're going to have very thin tunica media. In our smallest little venules and things, we're not going to have a complete tunica media. It's going to be an incomplete layer of smooth cells. So just kind of be aware of that. Think about that. Try and conceptualize it. That the closer you are to the heart, bringing blood to or away from the heart, the larger you are. And the further you get out there in the system, the smaller you are. And so if you're getting smaller, you're losing everything until you get down to the capillaries. And when you get to the capillaries, you're just a tunica intima with a small basement membrane. The arteries that take blood immediately away from the heart are conducting it away from the heart, and they are exposed to the greatest amount of force due to ventricular systole. So they have to be able to stretch. But then they also have to be able to bounce back so that we don't lose blood pressure during ventricular diastole and pass out. So those arteries are our elastic arteries, also called conducting arteries. And that's because they conduct blood away from the heart. They're called elastic arteries because they have a high proportion of elastic fibers throughout all of their tunics. And again, that's because they need to display the greatest amount of elasticity or stretch and recoil. So those are going to lead away into our muscular arteries. Muscular arteries distribute blood to a specific body region. So these are most of your named arteries, like your brachial artery, your radial artery, ulnar artery. Those are muscular arteries, and because they're distributing to a specific body region, we can also call those distributing arteries. So they distribute blood to a specific body region. And they're called muscular because relative to every other blood vessel type, their wall is the most muscular. So like 75% of a muscular artery's wall is smooth muscle. So this gives them the greatest impact on vasomotion. So about 75% of their wall is muscle. And so they have the greatest impact on blood pressure because of their greatest impact on vasomotion. Okay. The smallest of our arteries are the little arterioles that are going to lead into meta-arterioles in the capillary bed. So in our larger arterioles, the tunica media still has like 3 layers of smooth muscle cells. And when you get down to the smallest of the arterioles, you get like 1 layer of smooth muscle cells in the tunica media. And then when you lead into the meta-arteriole, it's like an incomplete layer. We've got these sphincters we'll talk about in a minute. But for arterioles, these have, by the time the blood gets to our arterioles, it's encountered the most amount of resistance it's going to in really an arterial path. And so we can also call these the resistance vessels. As all arteries are resistance vessels, sure, but as these are conducting arteries and these are distributing arteries, these are our resistance arteries. So our arterioles, the largest of our arterioles have 3 layers of smooth muscle in their tunica media, but the smallest have only 1 layer of smooth muscle. And they're going to lead into the meta-arterioles, which serves the capillary bed. So for our arterioles, we call them our resistance arteries. We could say they have 1 to 3 layers of smooth muscle in the tunica media. And these are going to empty, or they bring blood to meta-arterioles. What is this thing that I'm talking about? A meta-arteriole. So our little arterioles are going to come in and drain into these meta-arterioles that are going to lead into our capillary bed. And then our capillary bed is going to drain into a post-capillary venule. So here, we'll have our red arteriole is leading into our meta-arteriole, which is leading into our capillary bed. Our meta-arterioles have these little things called precapillary sphincters on them. Sphincters are just these little circular aggregates of smooth muscle that control the movement of materials from one place to another. And so if these precapillary sphincters are constricted, then the blood will flow right from the meta-arteriole into what's called the thoroughfare channel, and then that will drain into the postcapillary venule. So we can bypass the capillary bed. This is called a vascular shunt. And what happens here is we go straight from the meta-arteriole to the thoroughfare channel. And that will drain into our postcapillary venule. But we're not talking about that right now. We're talking about our arteries and arterioles. So these little meta-arterioles are going to be entering or allowing blood into our capillary bed, which is what we're going to talk about next. Smallest of our arteries are the arterioles that serve them. So here we go. Capillary. What is the structure of a capillary? Well, a capillary is just the tunica intima, which is also known as the endothelium. I forgot to bring that up on the first slide. And the reason I forgot to bring it up on the first slide is because I have actually been through this entire lecture once already today, 2.5 hours, but I had my microphone off. So it was like a silent film, and I'm recording again. So I can't remember what's the first and second time I told you. So here we go. What the heck am I talking about? The structure of our capillaries. Why? Because all they are is endothelium. Tunica intima is also called endothelium because it's the endothelium. It's within the blood vessel. It's the epithelial tissue lining the blood vessel. So for the structure of our capillary, it's just our endothelium and basement membrane. So we've got these simple squamous epithelial cells that are the endothelium. So there are simple squamous epithelial cells so that we can form the thinnest possible barrier across which we can have the diffusion of nutrients and gases. So the structure of a capillary is that it's just an endothelium with simple squamous epithelial tissue, small basement membrane. This endothelium is continuous with the endocardium of the heart. Okay. What's the function? To allow for nutrient and gas exchange. If we wanted to be really fancy, we could say it acts as a semi-permeable membrane against which blood can push. So, acts as a semi-permeable membrane. Okay, so what types of capillaries do we have? This one that I've drawn here is a continuous capillary. Continuous capillaries are the most abundant. So just be aware of that we find them all over the body and tissues all over the place, serving all the tissues. In places where you need like the blood-brain barrier or the blood-testes barrier, the endothelial cells are joined by tight junctions in continuous capillaries. In other places, the intercellular clefts are pretty small, but there's space between them, and you can find various types of cell junctions in different tissues. So continuous capillaries are the most abundant. What could we say? We have pretty small intercellular clefts. The next type we have are a little more permeable than continuous capillaries. Actually, probably pretty significantly more permeable. They're called fenestrated capillaries. Fenestra is the German word for window. And fenestrated capillaries have pores in their membranes. So when blood pushes against them and filtration happens, we can push out a little bit larger of molecules than we can get out just between the cells. So you can push things out, filter larger solutes through fenestrated capillaries. So we find these in areas where filtration is important, like kidneys where we're filtering your blood. We also find these in the small intestine where we need to do absorption of nutrients. So also bring small molecules into those cells pretty easily. So fenestrated capillaries contain pores, which are the fenestra, and in their membranes, and we find these where filtration is important, or absorption. So like in the small intestine, absorption of nutrients, in the kidneys, filtration of blood. Okay, well, what if we want to filter something out that's really big, like as big as a cell? We have to make blood cells. They've got to get out into the blood somehow, so we must have bigger kind of openings in some kind of capillary, and we do. We've got sinuses in sinusoidal capillaries, and we find sinusoidal capillaries in areas where we need to be able to filter or push out large solutes that are as big as even whole cells. So for sinusoidal, we'll say that we filter large solutes as large as whole cells. So we find these in our lymphatic structures, our bone marrow, which is a lymphatic structure, but your liver, your spleen. So your spleen stores blood, can't get in there from the bone marrow if we don't have big capillaries. If I wanted to add a little more here, I could say to this definition, I spoke it, but I didn't write it. So for our fenestrated capillaries, we could say this filters small solutes. So those are our types of capillary beds. Again, continuous is the most abundant, serving most of our tissues. Fenestrated has pores, so if I were to draw a fenestra up on this figure, then I would just see that I've got these holes in the plasma membrane. In my sinusoidal, those have the highest permeability, and if we look in them, not only do they have these really big openings in their plasma membrane, they have these really big intercellular clefts. And so that's what makes them leaky. It's like these sinuses. So if you're looking like an increasing permeability or increasing, permeability, then our continuous are the least permeable, our sinusoidal are the most permeable, so our permeability increases this way down the board. Okay. Capillary beds. So I've already kind of talked to you about the vascular shunt. This is what happens when you can constrict these precapillary sphincters on the meta-arteriole to allow blood to perfuse the capillary bed or to bypass it. So what's a vascular shunt? If here on our arterial side we constrict all of these precapillary sphincters, then we're going to shunt blood past this capillary bed straight into the thoroughfare channel. So for a vascular shunt, what we could say is that our precapillary sphincters are closed or constricted, and blood bypasses or is shunted past the capillary bed. We do not have enough blood in our body to fill all of our capillary beds at the same time. So because of that, we can only fill the capillary beds that need nutrient and gas exchange. How would we know which ones need nutrient and gas exchange? Well, if there's a lot of wastes out here, a lot of CO2 out here, then I need to serve this tissue. So in that case, I won't do the vascular shunt. I will relax my precapillary sphincter and flood or perfuse the capillary bed. If I'm good in nutrients and gas, or nutrients and oxygen, then I can just bypass this and go straight to the thoroughfare channel. So in a vascular shunt, we could say what we're doing is blood goes straight from the meta-arteriole to the thoroughfare channel, bypassing or shunting, bypassing the capillary bed. Okay, if the precapillary sphincters relax, then blood will perfuse the capillary bed and we will serve the tissue. So when the precapillary sphincters relax, blood perfuses the capillary bed and serves the tissue. There is yet another type of capillary bed where we don't have precapillary sphincters. Instead, how we control the blood flow into the capillary bed is by constriction or dilation of the meta-arteriole. So just be aware of that. So if this is my capillary bed and this is my meta-arteriole and this is my post-capillary venule, if this tissue needs nutrient and gas exchange, I can dilate my meta-arteriole and fill the capillary bed with blood. If I don't need nutrient and gas exchange, I can constrict my meta-arteriole and bypass the capillary bed. Okay. After our capillary bed, we're going to enter first into the post-capillary venule, and then those are going to merge together to form little venules that are going to merge together to form veins. So we have small veins, and in our small veins, starting at around 2 millimeters in diameter, we'll start to see the appearance of the valves in the tunica intima. And then they're going to converge together and become medium and large veins. We'll start seeing more and more smooth muscle as you get larger and larger, and more connective tissue as you get even larger still. So, and that's because the tunica externa is the thickest layer of a vein wall. So for our venules, we'll say these drain our post-capillary venules and converge to become small veins. So the post-capillary venule is a venule, but little venules will drain our post-capillary venules and converge to become small veins. In our small veins, we start to see valves in our tunica intima. And then we'll find them all the way as we continue converging and converging. As our small veins will converge to become our medium and large veins. In our largest veins, we still only have like 3 layers of smooth muscle in the tunica media. The tunica externa is by far the thickest layer. What are the largest veins? The inferior and superior vena cava. So they're going to be our largest, and they still don't have very thick tunica medias, but they have really thick tunica externas. So our small veins are going to converge to become medium and large veins, and we'll say even in our largest, our tunica media is only about 3 layers thick, and the tunica externa is the thickest. Okay. Another thing to be aware of when thinking of veins is that these are our capacitance vessels, which means they have a high capacity to hold blood. And the reason that that is the case is because there's not really any force pumping them through the system. Like when you get to the capillary bed, you disperse the force from ventricular systole, so you can really kind of have blood hang out in venous circulation, and then when you need it, get it back to the heart. How do we do that? Well, we squeeze on veins. Our skeletal muscles contract and we squeeze on veins, and then it returns more blood to the heart. Then we can get more blood out into the system and increase cardiac output, so that as you need more blood, you get more blood. And we can do that by storing it here in the veins. So they call veins a blood reservoir. Why? What do reservoirs do? They store things. So they store blood. And so this is what makes them the capacitance vessels, is that they have a high capacity to store blood. So if you look at 100% of your blood volume at any given moment, well, not 110%. You might try and give 110%, but you've only got 100%. So if you have 100% of your blood, about 55 to 60% at any given moment is in your systemic veins and venules. Systemic veins and venules. And then about 15% is in your systemic arteries and arterioles. About 12% is in your pulmonary circuit. About 8% is in your heart, and about 5% is in your capillaries. When you look at this distribution of blood, I don't know, when I look at it, it blows my mind. Capillaries is where we're serving our tissues, and at any given moment, only about 5% of our blood is in them, which means that most of our capillary beds at any given moment are being bypassed. Most of our blood at any given moment is hanging out in our systemic veins and venules. So if we have our 55 to 60%, we've got our 20% here, 20% here, 40% plus 60%, 100% of blood. Most of that is in our veins, which is pretty cool. We will pick up with physiology next time. What I want you to think about when you're working on your lab or thinking about like the anatomy that you need to worry about, you need to think about histologically how our veins and arteries different. And then I want you to be able to follow blood pathways through the heart and then through the body. So you already know the pathway of blood through the heart, but now could you follow it out to a specific part in your body? So look at your lab guide, and anything that's bold or it says that you're required to know in your lab objectives, you have to know. And you have to know an organ served by that. So if you feel like it's been a short lecture and you want more time, then I would say it's going to be a long lab, because you need to be able to follow blood out of the heart through all of the vessels that you have to memorize and tell me a body part that they're serving. So, and then, I don't know, potentially even follow a pathway of blood to all the way from the heart to like your big toe and back. And tell me all of the structures that you would encounter. So I want you to work on that when we leave class today. So first, thinking about, though, what are we going to think about? Our anatomical differences. Histologically. So when you're looking at your slides, you'll always be able to tell an artery and a vein apart, because a vein, even the biggest vein, never has a tunica media that's really thick. And an artery always has a tunica media that's really thick. So thick, in fact, that when arteries are empty, they can still maintain their shape. That's not the case with veins. When veins are empty, they collapse. So just be aware of that. And also in veins, their tunica externa is really, really thick. So you should be able to recognize that difference histologically. So be able to note that when you're looking at your slides. The difference between artery and veins, how you can tell which one is which. Look at all the layers of their wall. And then when you're thinking about other parts of anatomy, start at the heart. You already know blood flow through the heart, but now continue from your aorta through your systemic circuit. Follow that brachiocephalic trunk to your right arm and up the right side of your head. And then from on the left side of that aortic arch, we have our left common carotid artery and our left subclavian artery that go out to the left arm. On the right side, the heart is sided. On the right side, we have a brachiocephalic trunk that comes off of it, and then we've got our right common carotid artery and our right subclavian. Follow that down the left arm. Superior to the diaphragm, the descending aorta is called your thoracic aorta, or your descending thoracic aorta. So check that out. Inferior to the diaphragm is your descending abdominal aorta. And you need to know some branches. You need to know the celiac trunk. What does it branch into? Who does it serve? You need to know the superior mesenteric artery. You need to know the inferior mesenteric artery. Our aorta is going to branch into the common iliac arteries that are going to branch into the external and internal iliac arteries that are going to branch into the femoral artery. That's going to give rise, the femoral arteries are going to give rise to the popliteal arteries and the posterior and anterior tibial arteries. You need to know all of those things. So follow your way out. When you get there to the tibial artery, if you were dropping off oxygen, what would you go through? An arteriole, a meta-arteriole, the capillary bed, a thoroughfare channel, a post-capillary venule. What if I was coming back to the heart? Then I'd go through a small vein, a medium vein, a large vein, and ultimately return through my, if I'm coming from the leg, I'd come back through my inferior vena cava. So could you do that? Could you draw it? You should. So try that. So I'm going to actually leave that to you for your homework in lab right now. So that was most of the arterial path that you need to worry about. Could you finish that path? Could you then add on your companion vessels, the veins that are going to run with them? That's your challenge. We'll pick up next time talking about blood flow. This is why we care. Our tissues need blood flow. So have a look at the relationship between blood flow, or the flow of anything for that matter, to pressure and resistance, and we'll pick up there next time. Okay, so cut back to arterioles. Arterioles.